The structure and function of DNA. DNA has two functions, an autocatalytic function and a heterocatalytic function. Its autocatalytic function is to direct its own synthesis. Its heterocatalytic function is to direct the synthesis of RNA and proteins. There are 4 types of organic molecules necessary to support life. These are proteins, lipids, carbohydrates, and nucleic acids. Nucleic acids are the building blocks for both DNA and RNA in living organisms. DNA is composed of a string of nucleic acids. An individual nucleic acid is composed of a phosphate group, a ribose sugar, and a nitrogenous base. There are only 4 nitrogenous bases found in DNA. These are adenine, guanine, cytosine, and thymine. RNA and DNA nucleotides both contain a phosphate group. RNA and DNA nucleotides both contain a sugar molecule. DNA nucleotides have a deoxyribose sugar molecule. This is why DNA stands for deoxyribonucleic acid. In RNA, the sugar type is a ribose. This is why RNA stands for ribonucleic acid. The 4 nitrogenous bases in RNA differ in the fact that instead of thymine, we have uracil. So the 4 nitrogenous bases in RNA are adenine, cytosine, guanine, and uracil. In a molecule of DNA or RNA, individual nucleotides are linked together to make a sugar phosphate backbone. Each strand of DNA is connected to the other DNA strand through hydrogen bonding. This hydrogen bonding occurs between the nitrogenous bases on opposing DNA strands. There are base pairing rules in DNA. A can only pair with T. This bonding creates 2 hydrogen bonds. And C can only pair with G, which creates 3 hydrogen bonds. The DNA strands are oriented anti-parallel to one another. The ends of the DNA molecule are different. One end of the DNA molecule is designated 3' and the other as 5'. They are named according to the carbons in the ribose sugar molecule of the DNA backbone. In a molecule of DNA, one strand will be oriented from the 3' to 5' direction, and the other from the 5' to 3' direction. Watson and Crick began trying to construct a double helix that conformed to all of the characteristics known about DNA. This idea of specific base pairing was a flash of inspiration that enabled Watson and Crick to solve the DNA puzzle. A pairs with T and G pairs with C. A is also said to be complementary to T, and G being complementary to C. When a cell reproduces, it must duplicate its DNA, providing one copy to the new offspring cell while keeping one copy for itself. If you know the sequence of bases in one strand of the double helix, you can very easily determine the sequence of bases in the other strand by applying these base pairing rules. The 2 strands of parental DNA separate, and each becomes a template for the assembly of a complementary strand from a supply of free nucleotides. The nucleotides are lined up one at a time along the template strand in accordance with the base pairing rules. Enzymes then link the nucleotides to form new DNA strands. The completed new molecules are identical to the parent molecule and are known as daughter DNA molecules. The process of DNA replication requires the cooperation of more than a dozen enzymes and other proteins. DNA polymerases are the enzymes that make the covalent bonds between the nucleotides of a new DNA strand. As an incoming nucleotide base pairs with its complement on the template strand, a DNA polymerase adds it to the end of the growing daughter strand. DNA replication begins on a double helix at specific sites called origins of replication. From here, replication then proceeds in both directions, creating what is called a replication bubble. The parental DNA strand opens up as daughter strands elongate on both sides of each bubble. The DNA molecule of a typical eukaryotic chromosome has many origins, where replication can start simultaneously, shortening the total time needed for the process. Eventually, all the bubbles merge, yielding two completed double-stranded daughter DNA molecules. The molecular chain of command is from DNA in the nucleus to RNA to protein synthesis in the cytoplasm. The two stages of gene expression are transcription and translation. Transcription is the transfer of genetic information from DNA to an RNA molecule, which is messenger RNA or mRNA. Translation is the transfer of the information from the mRNA molecule into a sequence of amino acids, forming a polypeptide or protein strand. In DNA, the monomers are the four types of nucleotides, which differ in their nitrogenous bases, A, T, C, and G. The same is true for RNA, although it has the base uracil instead of thymine. There are only four different types of nucleotides. In DNA, they are A, G, C, T. In RNA, it's A, G, C, U. During translation, triplets of bases called codons will specify for 20 amino acids. There are enough triplets or codons to allow more than one coding for each amino acid. For example, the base triplets AAA and AAG both code for the same amino acid. The genetic code is a set of rules that converts a nucleotide sequence in RNA to an amino acid sequence. 61 of the 64 triplets code for amino acids. The triplet AUG has a dual function. It codes for the amino acid methionine and is also the signal to start the polypeptide chain. Thus, each polypeptide chain begins with methionine. 3 codons, UAA, UAG, and UGA, do not designate amino acids. They are stop codons that instruct the ribosomes to end the polypeptide. In transcription, the two DNA strands must first separate from one another at the place where the process will start. Only one of the DNA strands serves as a template for the newly forming RNA molecule. The other strand is unused. The nucleotides that make up the new RNA molecule take their place one at a time along the DNA template, forming hydrogen bonds with the nucleotide bases there. Notice the RNA nucleotides follow the usual base pairing rules, except that U, rather than T, will pair with A. The RNA nucleotides are linked by the transcription enzyme RNA polymerase. The signal to start transcription is a nucleotide sequence called a promoter. The promoter is located in the DNA at the beginning of the gene. A promoter is a specific place where RNA polymerase attaches. The first phase of transcription, called initiation, is the attachment of this RNA polymerase to the promoter and the start of RNA synthesis. For any gene, the promoter dictates which of the two DNA strands is to be transcribed. During the second phase of transcription, called elongation, the RNA grows longer. As RNA synthesis continues, the RNA strand peels away from its DNA template, allowing the two separated DNA strands to come back together in the region already transcribed. In the third phase, called termination, the RNA polymerase reaches a special sequence of bases in the DNA template called a terminator sequence. This sequence signals the end of the gene. At this point, the polymerase molecule detaches from the RNA molecule, and the DNA strands rejoin. The eukaryotic cell not only localizes transcription in the nucleus, but also modifies or processes the RNA transcripts there before they move to the cytoplasm for translation by the ribosomes. One kind of RNA processing is the addition of extra nucleotides. These nucleotides are placed at the ends of the RNA transcript and are called the cap and tail. The cap and tail mark the mRNA for export from the nucleus and helps ribosomes recognize the RNA as messenger RNA. Another type of RNA processing removes non-coding regions called introns. The exons are the coding regions, which are the parts of the gene that are expressed. Both exons and introns are transcribed from DNA into mRNA. But before RNA leaves the nucleus, the introns are removed and the exons are joined to produce a messenger RNA molecule with a continuous coding sequence. This cutting and pasting process is called RNA splicing. The first important ingredient required for translation is messenger RNA. Once the final product of messenger RNA is present, the machinery is used to translate mRNA requires enzymes and sources of chemical energy such as ATP. In addition, translation requires ribosomes and transfer RNA. At one end of the folded molecule is a special triplet of bases called an anticodon. The anticodon is complementary to a codon triplet on the mRNA. During translation, the anticodon on the tRNA recognizes a particular codon on the mRNA using base pairing rules. At the other end of the tRNA molecule is a site where one specific kind of amino acid can attach. Although all tRNA molecules are similar, there are slightly different versions of tRNA for each of the 20 amino acids. The subunits of the ribosome act like a vise, holding the tRNA and mRNA molecules in close proximity. The ribosome can then connect the amino acid from the tRNA to the growing polypeptide. Initiation of translation begins with step 1. In step 1, an mRNA molecule binds to a small ribosomal subunit. A special initiator, tRNA, then binds to the start codon where translation is to begin on the mRNA. The initiator tRNA carries the amino acid methionine, and its anticodon, UAC, binds to the start codon, AUG. Step 2. A large ribosomal subunit binds to the small one, creating a functional ribosome. Next, we have elongation. Step 1 of elongation is when the anticodon of an incoming tRNA molecule carrying its amino acid pairs with the mRNA codon at the A site of the ribosome. In step 2, the polypeptide leaves the tRNA in the P site and attaches to the amino acid on the transfer RNA in the A site. The ribosome creates a new peptide bond. Now, the chain has one more amino acid codon in the A site of the ribosome. In step 3, the P site tRNA now leaves the ribosome, and the ribosome moves the remaining transfer RNA carrying the growing polypeptide to the P site. The mRNA and tRNA move as a unit. This movement brings the next mRNA codon to the A site in order to be translated, and the process can start again with step 1. Termination begins when the stop codon reaches the ribosome's A site. Stop codons, UAA, UAG, and UGA, tell the translation to stop. The completed polypeptide is then freed, and the ribosome splits back into its subunits. Sickle cell disease can be traced to a change in a single amino acid in one of the polypeptides in the hemoglobin protein. Any change to the genetic information of a cell or a virus is called a mutation. Mutations can involve large regions of a chromosome or just a single nucleotide pair, such as in sickle cell disease. Occasionally, a base substitution leads to an improved protein or one with new capabilities that enhance the success of the mutant organism and its descendants. Much more often, though, mutations are harmful. Mutations within a gene can be divided into two general categories: nucleotide substitutions and nucleotide insertions or deletions. A substitution mutation is the replacement of one nucleotide and its base pairing partner with another nucleotide pair. Because the genetic code is redundant, some substitution mutations will have no effect at all. For example, if a mutation causes an mRNA codon to change from GAA to GAG, there will be no change in the protein product. This is because GAA and GAG both code for the same amino acid, glutamine. These types of mutations are called silent mutations. Other substitutions involving a single nucleotide do change one amino acid to another. These mutations are called missense mutations. For example, if a mutation causes an mRNA codon to change from GGC to AGC, the resulting protein will have serine instead of glycine at this position. Because mRNA is read as a series of triplets during translation, adding or subtracting nucleotides may alter the triplet grouping of the genetic message. These types of mutations are called frameshift mutations. Mutations can occur in a number of ways. Spontaneous mutations result from random errors during DNA replication or recombination. Physical and chemical agents called mutagens can also cause mutations. These include high-energy radiation, like X-rays and ultraviolet rays, and chemical mutations such as toxins and pollutants. Thank you for watching.